Thanks, Neil. So clearly, in the development process of biomarkers, as we've seen from the 5 talks, that there are multiple challenges in every step of the way, from the development to deciding on the test itself or the evidence of that test. But also others is how to transform from a monoanalyte to multiplex, and what do we do about the LDTs. So I'm going to call the 5 speakers to come up to the table, and then we can talk. We can take questions from audience and have discussions. So the floor is open for any question, discussion, or suggestion. Yes, Rich.
Good morning, Rich Shilsky from ASCO. So I wanted to start, I guess, with a question that Mia raised, but I'll address it to the whole panel, and that is the issue of variant annotation. You know, there are lots of knowledge bases being developed. There are publicly available knowledge bases. There are commercially available knowledge bases. You know, every entity that's offering NGS testing is developing a knowledge base. It's not clear, you know, that there are any established criteria, you know, for how to establish a knowledge base, for how to call specific variants. And it's also not clear that any of this information is being shared. And I wonder if the panel feels that this is sort of a productive exercise to continue, and whether or not, you know, perhaps a recommendation that could come out of this workshop, you know, would be to call on, you know, a public agency, could be the NIH, I suppose, could be the National Library of Medicine, could be some sort of overarching entity that would develop, you know, this variant annotation process and database for the community as a public good, and would, you know, curate it and keep it updated and so on. So just curious to get some thoughts about that.
Mickey or Mia, would you like to start?
Thank you, Rich, for that question. So I just showed you one example of our effort at Vanderbilt with My Cancer Genome. And there are, as Rich alluded to, there's a number of different other annotation efforts, both at other academic medical centers and other private groups. And I think that what we're seeing is that in the germline space, the federal government, specifically the NCI, I'm sorry, the NIH and the NHGRI have stepped up to do this effort on the germline side. There are ClinVar and ClinGen, both efforts to both collect clinical variant raw data collected with phenotypes, as well as annotate on top of those to make publicly available resources. And I think they are starting to develop a process for the workflow needed to validate those curation efforts, and that is a great model to start with. Their models, unfortunately, are not entirely conducive to cancer in the sense that we require a much deeper clinical outcome data than just an initial phenotype. So our challenges are different, but I think we could build upon those efforts very nicely if there was a focused funding source, and I believe when I have spoken to those other organizations, they have said, well, the 800-pound gorilla of the NCI should be the one that should step up and do that. So, you know, they're focusing their efforts in germline, and until we get some focused funding to say we're going to move forward with this, I think these individual efforts will stay in their silos, which is an unfortunate reality.
I think the NIH is losing weight. It's probably a 700-pound gorilla now. So.
Mickey, would like to add anything? Would you?
Thank you. I was hoping that ASCO and NCCN would step up too, but I think the feeling is shared that the time is now. Everybody's acting on information, and if we could have some common data that everybody could point to so that we knew we were acting identically across sites as we get this information, it's extremely important, and the time is now.
I want to make 2 points on this. Number one is that there's 2 aspects, I think, of this that are somewhat of a concern. Number one is if you take a look at the validation steps used in the Foundation one, and just so you know, I don't have any financial interest in in foundation medicine. But when you take a look at their validation, most of their validation was done on pools of cell lines and then diluting out the cell line DNA with something else. And the amount of validation they did in their assay on actual tumor specimens, not surprisingly because they're relatively precious, is very few. They actually, in their publication, only did 3 colon cancers where they had, and did it on more than 70 successive days and got the same result from that one huge, or from 2 very large colon specimens. And so I think when we think about validation, I think we need to hold ourselves to the standard from the actual one. The second thing is that, you know, almost all the relationships between initially between the response rates and outcomes were done from relatively enriched specimens, and that, you know, the relationship between the clinical response and the findings. And now that we go to more and more sensitive platforms, are we going to be seeing the same relationships? And the clinical associations of that, I think, need to be looked at a little bit more carefully as they ratchet up the sensitivity of these and are picking it up in ones where there's less and less of the tumor specimen of the mutation able to be detected with there.
I also wanted to add another point beyond just the annotation databases or the databases themselves of the actual results. You know, we're looking at, we really want to avoid a myriad number 2 again. You know, they have the largest collection for decades of BRCA mutations in germline for breast cancer risk and ovarian cancer risk. And no one else can get access to that database. And there's been efforts like Free the Data, where women are actually uploading their own BRCA report results up to available resources, and they're manually transcribing it in order to just be able to liberate this data. So your point about Foundation, they now have the largest database of tested samples. That doesn't mean they have the clinical annotations to go along with it, but we do need a new paradigm in order even just to get the data liberated.
So I want to advocate for the big gorilla, because I think what we have learned over the last 10, 20 years is that the amount of knowledge is just exponentially growing. And this will probably continue. And if we were to choose the most unbiased and hopefully most long-lasting institution that there is, that might be a government institution. So if there was a mechanism, funding mechanism for accumulation of data that could not be a chain of different databases that pop out here and there, but one major database that will last for the next, let's say, 100 years, and that should be really the goal. I think that would be worth the effort, and it will outlast everybody's individual career. Of course, everybody wants to have their own database, and I would love to have one, but I think it's a public trust that we have to really respond to.
Okay, any other issues? Yes.
Mike Caligari, Ohio State University.
It's okay.
Oh, I'm sorry.
Go ahead.
Mike, can I?
Please.
It's okay.
Please.
Okay, so I guess there was once a time in medicine where physicians were actually able to use clinical judgment. Unfortunately, what's happened is we've had what's called evidence-based medicine become somewhat of a dictate. And there was a lot of good reasons to do that. So to make sure that physicians really were following what was the best possible practices and trying to standardize across many physicians, there was this term generated called evidence-based medicine, and people wanted to say, I'm performing evidence-based medicine. Now, the downside of that is what I think we're seeing now with molecular diagnostics, where the use of this term evidence-based medicine is preventing us from doing what I think everybody in the field thinks is the right thing to do, which is to molecularly profile every single patient who has at least advanced cancer and potentially move that forward to everyone who has early stage cancer. And this term evidence-based medicine is used as an excuse to not pay for that type of diagnostic testing. But there's another way to think about this, which is if you had a parent or a sibling who had cancer, what would you do for that person? What would you do for your dad? I get that that question all the time in my clinic. And the question here is there actually anybody on the panel if they had a close family member, a sibling, a father, a mother, a child who had advanced cancer, would they actually not want to get this type of molecular testing, given all the caveats we've heard about in terms of variability of the assays and issues of sensitivity and specificity? Is there anyone on the panel who actually would not want to do that or is not currently planning on doing that if they have a family member? And if there isn't, then really what we're getting down to in terms of this whole debate over 2 days is that we don't have sufficient funding to pay for this, or at least insurers will not. And so we're using guidelines and evidence-based medicine as an excuse not to do this for at least some patients, even though it's going to get done for people who have sufficient funds to personally pay for it, or people who have sufficient connections to get into a program where it's being done for free.
Is this a family member we're getting along with?
So anyone would like to address?
I would order this for my family members and even ones I didn't get along with that well.
So thank you for that, and I'm very sympathetic. I don't think the issue is whether or not we should be doing this. It's more how, how do we do this? And I think there have to be some rules. And as soon as you start putting any rules down, they become unpalatable, unfortunately. And so I think we need a more palatable set of rules than we currently have. And what I think, one thing that we have happening right now is different people interpreting the same rules differently. So at my institution, for example, we run exactly the same assays or very similar to what you run and what Foundation Medicine runs. And my pathologist simply will not issue a report. They're like, this is not validated. There's no predicate instrument. There's, this, that, and the other thing. Your institution feels comfortable doing it. Foundation Medicine feels comfortable doing it. I mean, what are the rules going to be here? And one more minor thing is, I think on the sequencing side, I'm feeling pretty good about these assays. I think most of the single nucleotide variants and small indels we're calling are real. But I can tell you right now, the copy number alterations that are coming back on the Foundation Medicine assay and our own assays have a much lower level of validation. So what I ordered for my family member, absolutely. But I know, because I've done 1,400 of them, that if I saw a copy number alteration in chromosome 3q and they said that that's a PIK3CA amplification, I would know whether I believe that or not. If they saw, a canonical activating mutation in PIK3CA, I would believe that. So we still got to get the ground, there's still some real issues here. So I'll leave it at that.
So I would say it depends on the family. If your son-in-law is Neil Hayes, I think you're lucked out. But it's also that certainly it's very attractive for patients who don't have family members who are savvy or who don't have an oncologist who's interested in really going through these data, that we get this pile of results back, and it can be highly confusing, can be upsetting, because all of a sudden there's some hope that there might be these agents that are on the list on Foundation 1 as an example, and they might not be useful. So I think, would I order this for a family member? Yes. But only if there was truly somebody who could interpret the data, and if there was a discussion before ordering these tests that the expectations should be realistic.
I would add to this, actually, because of the variety and the presence of multiple of those tests, as we've mentioned, I would totally concur. It's not only resources. I mean, rules has to be put for specific resources, because at the end, it's going to be very difficult to just do 100 tests or 100 different tests for a patient at one time. I think also the question is, what do you do with the data, and what kind of further therapeutics you're going to be using that could be actually harmful if it's not going to be done properly.
So Mike Caligari from Ohio State University. Neil brought this point up for anybody on the panel, and that's the issues of patents, because at least in drug development, if you got a great drug and it shows great activity and your lawyers tell you we can't get our arms around a patent, that drug may never see a patient. Is that going to be the same, with the DNA and RNA technology, where it's going to be so muddled that even if we have something that can, in fact, be useful for clinical decision-making, the law will actually prohibit any company from really taking it forward so that there can be a charge?
I think, we just got a patent issued. I think VC certainly still wants to see a patent. You need money to go forward. So I think there's sort of a path forward for the moment, but it's much more expensive than it should be. It's going to inhibit So our tech transfer office, for some reason, supported us in an endeavor that's, like my cancer center director told me the day I said, the tech transfer office took on my patent, he said, you'll never see a dime. And, it's just very low enthusiasm for moving diagnostic patents forward. I think anyone who's involved would share that. And I think it's an important issue. I'm not an expert, but there is a path forward. I think it's important. We need more discussion about it. Maybe we'll hear more about that this afternoon.
And the other side of this, Michael, is actually going back to the evidence or the strength of those assays. Because of the lack of sharing between different companies that are trying to develop those assays for the same purpose that you just mentioned, is that it's becoming even more difficult to develop a unified assays or assays that can be dependable for further development of companion diagnostics or otherwise. Yeah.
As a person who, I'm one of the people that gets revenue from post-marketing royalties from EGFR mutation testing, and we didn't see too much of it for a while, but in 2009 it started to kick in. Now, one of the things that happens from an enterprise-wide effort is that if somebody holds the intellectual property for giving a specific class of drugs, which it was in this particular one, because the test was linked of a genomic change to giving a class of drugs, is that people end up needing to license that technology. So if you get a multiplex platform, you're going to have to go to multiple patent holders who have the link between classes of drugs and specific tests, which, although it's nice individually to reap the benefits of that, from a practical standpoint, it becomes quite a challenge when you are going to end up like in lung, where we likely will have at least 5 and probably somewhere to 6 to 10 different genomic changes that indicate specific drugs.
And just say the inconvenience, the cost would also go up of those assays because you're paying royalties to the other groups.
Well,
So
I don't think it's so bad that the costs go up for the inventors. So.
Sure.
Hi, Federico Monzon from Invite Corporation. We're a diagnostic genetics reference laboratory, and I think I'm here standing as somebody from the molecular diagnostics community, addressing one of the questions that Miki came up, but that some other speakers also kind of talked about. His question was, are we all doing the same thing? Are we all getting the same results from at least next generation sequencing assays? And I think we definitely are. Implementation of clinical tests is usually done by accredited laboratories. Most of us have College of American Pathologists accreditation, and that requires us to really, really look at the quality of our systems, look at the reproducibility of our results, and actually do something that is called proficiency testing, which means we need to make sure that we get the results and that we're comparable to other laboratories, that we interchange samples with other laboratories. Some of these samples come directly from large collegiate organizations like the CAP, College of American Pathologists, and some of them we do interchange between the laboratories if they're not available. And for the most part, we are getting the right results. So I think, I want to a little bit dispel the notion that it's a wild west out there of diagnostic testing. It is not. We are a regulated environment, and we act very responsibly because every one of our genetics laboratories acts on behalf of the patient. We have the patient, in the first of mind, and that's one of the things that I want to be clear. Are there issues? Are there things, like Neil mentioned about, copy number and, the variability in how laboratories address that? Yes, there are issues that need to be solved. But for the majority of the clinically available tests right now, I think the robustness and of the technology plus the effort that the laboratories put on clinical validation and on ensuring that those results are accurate, reproducible, speak for themselves. There is the question of clinical validity, and that's a different thing, right? Currently, the regulations do not necessarily establish that we need to have clinical validity in order to offer a diagnostic test. And that's where, I would say there is a little bit of, necessity to clarify the rules. I think Neil talked a little bit about the issue of LDTs and whether FDA should step in or not. I'm going to talk tomorrow, so I'll probably leave that for later. But let's just say that, the drug mentality of, how do you monitor a drug and how do you go applying regulation for drug development and, getting a drug out there for use for patients does not necessarily apply for diagnostics. And we need a different framework that is, right now being discussed, and hopefully we'll come to an agreement on how we do these more responsibly.
Now, I'm sure we'll talk about this more in the next session with regulatory, but as you very well know, one of the most common questions as medical oncologists we get is, I got my CA19-9, what do I do with it? And when you think about the numbers of assays that's going to happen without that clinical qualifications or validity, this is where a major issue that has to be addressed and put forward, because yes, companies can do those kind of things. And you're right, everyone wants to do for the best of their patients, but also stakeholders. So there are things that has to be addressed with this that are extremely important. Mia?
I think one of the other components to add is that Mickey alluded to how difficult it is to validate these assays. And not all of the variants or not all the genes that are tested for in these assays will be relevant to every single cancer as part of standard of care. And yet you don't want to have to revalidate these assays for each individual cancer type. Now, that doesn't mean that, and what some diagnostic companies are doing is they're actually saying, well, I'm going to run it, but if you ask me, I'll only report and only charge you for the ones that you ordered that might be relevant to a particular cancer type. And that way the clinician's also not getting overwhelmed with information that they don't know what to do with, but you're having a single analytic validation pipeline that you can mix and match into different commercial products that you offer. So I think there's some creativity along this lines that's allowing for flexibility in the marketplace.
I think Roy, jump in real quick, just to go back to Frederico's comment. I'm not so certain I agree with what you said. I do feel that we are somewhat in the Wild West, and I'll just give you a for instance. As far as sequencing in tumors, if we aren't stating what our tumor content is, my lab could be sequencing 20%. I know a commercial company that will take 20% or greater. Another lab may be using a greater tumor content. If we're dealing with heterozygous or even a mutation that's not a 50%, there's no guarantee that everybody's going to see that variant necessarily and see it as reproducibly. And the other thing is the proficiency testing is pretty low bar. I would say, number one, we should maybe make it a little bit more stringent, and I would love to see it public, to see how well labs are actually doing in this, because it's not 100% accuracy that you need to demonstrate in order to get by with the proficiency test. So I think having some good reference materials that everybody could be testing would be a good step in the right direction. I don't think it's tremendously bad. I think everybody's going to see a KRAS, everybody's going to see a BRAF. But some of these variants that we're seeing that we don't know the annotation, that are popping up in 1 or 2 patients in COSMIC, what do we do with those, and how do we know that everybody's seeing that? We don't.
One of the other examples I'd like to give in this that I think points out one of the problems is that there's MET amplification in lung cancer de novo that's present in about 1 to 2% of patients. And depending on the call rate, it turns out that if you end up looking at this, it's really only very active in ones that have a DNA, an actual amplification, not polysomy, of greater than 5-fold. And at least Foundation reports that they're not very accurate when you get below 8 copies. And so when you get a report back that it's MET amplified and you don't know the quantitation of it, becomes potentially very important as a predictive marker for efficacy of crizotinib, one of the MET inhibitors.
Let me follow up on that, because having again looked at hundreds and hundreds of these on clinical patients, even something as important as HER2. If you look at the genome of HER2 and you look, because we actually, in our lab, we never report the next gen results. We always will do a second validation. That's what our lab makes us do. And HER2 is a much more complicated locus than you would ever think by FISH or by immunohistochemistry. And it's almost back to the drawing board on HER2, with indels and mutations and other things. So, MET's just focal high-level amplification, but, on top of polysomy, HER2 is very complex, and I think, it is sort of back to the drawing board on what is really abnormal. So there's a lot of fun stuff, but it's also, we would never have called it a alteration by a FISH assay or immunohistochemistry, but it's clearly abnormal by next-gen.
Mia, I'd like to just start taking other questions because some people are still standing. Please.
Yes, Roy Herbst from Yale. I want to bring up a point and also ask a question of the panel regarding the future of the companion diagnostic. And the issue I want to bring up is we focused a lot on DNA and RNA-based tests, but protein-based tests are still there. And the biggest example right now, I think, is the immune therapies and this whole aspect of this PDL1, which is an elusive marker, but it is being used. And we're in the situation now where 5, 6 companies are developing immune checkpoint inhibitors, each with a different assay. And if this moves forward, this assay is even more complicated than what we're discussing here because the cutoffs can be different, how you test, where you test, and so forth. So I guess, again, it's a very open-ended question, but one, the question is there anything that can be done to try to coordinate this early on? And is the companion diagnostic dictum, is that making this even more complicated?
This is a question that we had yesterday, actually, at SITC, and we have a biomarker working group, small steering committee, but you're right. First of all, it's 8 companies, not even 5 and 6. But the issue is, yes, none of them can match the other, and actually lots of trials, as you very well know now, when they even compare between one or the other, it doesn't really match. Some of them is positive, some of them is negative. And the joke is you continue doing it and it's positive and then you treat the patient. But it's an issue, it's a problem. And one of the major issues relevant to this is what I was talking to Michael about, is the issue of sharing those kind of data prior to actually getting it out. Now, the pre-competitive space here is extremely narrow, so there is no leeway for companies to do that. Now, we're making progress because even now that we broke a major ceiling where companies are putting together different drugs in clinical trials. So that actually is an example and a model that can be done and can be implemented for this kind of data sharing. But it's always going to be dependent on the regulation and on the incentives, either from Congress or from FDA. Other than that, it would be extremely difficult to actually put that in.
Well, I'd just like to say that it's almost unconscionable that we're using immunohistochemistry in 2014. I mean, I actually think that's almost disingenuous, and I think that I almost think that the companies would love a bad biomarker. And, if they get a kind of okay one, that's fine too. But, I think that we should be using more robust technologies than immunohistochemistry, I mean, including RNA-seq.
I actually disagree with that in that there's at least within the science of this worrying about whether the PD-L1 needs to be in the tissue macrophages versus the tumors, and there aren't those particular tools don't lend themselves real well to that sort of situation until it's worked out. I think it's inappropriate to be doing the test to help working it out. Now, the other technologies may supersede it, but for the current time, I think it is an important piece.
Yeah, and I would agree with that because it depends on the molecule and the drug that you're using, basically.
I mean, immunohistochemistry. You just said it. There are 8 different tests. They all disagree. I mean, what is the evidence that they really know the epitopes that they're targeting here? I mean,
See, I just wanted to demonstrate the challenges that we're having.
But I have to overstate my case because that's why I'm here.
Please.
No, but I think one way to figure out, I think the key problem is the Wild West inspires ideas. It's a competitive market, and certainly there's a big reward, a lot of money, and so forth. On the other side, if you have 8 different tests that are all reasonably good, all different, who should in the end be the winner? Should we combine this or not? Should it be the free market that defines who is the winner, which might not scientifically be the best? Or should there be a stronger regulatory body or a jury for these markets that defines which test is being done? So the studies, as has been said before, that are needed are to do a prospective study, compare different commercially available tests at the same time. And it's, of course, difficult to do because there are financial interests for each individual test. But if you had 8 markers that are simultaneously tested in the same patient cohort, you might find out which marker is better than the other. But you need a regulatory body to tell the Wild West what to do.
Please.
Lisa McShane, NCI. My question follows up on this discussion about the Wild West and the variation in the different assays. So for those like Mia who are building knowledge bases, and some would like to see NCI build those, how do you cope with capturing that kind of data that describes the differences between assay methodologies, or even, bioinformatics pipelines could be versioned a couple of times a year for NGS platforms? If we're really going to start sorting this out, we'd have to capture that information, and it just seems almost impossible to me to capture what I'd really like to have captured. So how are you dealing with that, Mia, or anybody else in the room? And
That's kind of where I was going with my suggestion, that we need to capture minimal information about the assay, how it was done, so that it could be reproduced. In other words, do you report on 20-fold read depth, 30-fold read depth? What's your minimal coverage? What is your wet lab kit that you're using, and the pipeline that you're using for analysis? If we could begin to capture that and come up with the minimal information that everybody needs to put in to their data when they publish, if nothing else. Heaven forbid we'd start a database and not capture that, because it could be then very difficult to
Right, I agree. So I was just wondering, what of those who are already curating these databases, what are you capturing, and what do you think is feasible to capture?
I am not curating an NGS database right now as far as
No,
our
but
content.
it even extends to other assays. I mean, if you're putting out a report saying that, if you have copy number alteration in a certain gene, if you've done different assays to do that, is that kind of information being captured and conveyed in the report that gets put out from one of these systems?
So as part of the reports and I'm not a molecular pathologist, I'm an oncologist, what I see is they do explain their assay as far as the kit that they're using, whether it's for IHC or FISH or any other types of assay that they're using. So that comes to us as part of the interpretive report, which helps us understand the analytic validity of the assay that they're giving. But it also is going to depend, as far as these databases are concerned, which files and who's contributing the data, because as the oncologist receiving this data, all I'll get would be a variant report. I don't get any of the BAM files or the VCF files. That's all residing still within the hands of the molecular diagnostics lab. Whether or not they'd be willing to share that, this is really a, whose authority and responsibility is it to share that data? And certainly on the receiving end of the molecular pathology report, as the physician, I would have absolutely no insight into the subtleties of the percent of tumor content and the thresholds that any individual company was doing. So it would need to be a collaboration between both the molecular diagnostics labs as well as the clinical annotations. And that is just strategically a very challenging issue.
Within the Lung Cancer Mutation Consortium, a group of 16 centers across the United States, they're actually assembling the BAM files and are going to be comparing them across the institutes, and they will have an effort to see how reproducible they are, not just across the reads, number one, and then number two is sharing some samples. Now, the interesting thing is that the group did this with the earlier technologies. Most of them are using snapshot or direct DNA sequencing from the publication in JAMA this year. On that one, they have had a very difficult time getting it published. Nobody is very interested in publishing the ability to reproduce the calls of genetic sequencing within lung cancer. And so we're going to our specialty journal, the Journal of Thoracic Oncology, because the level of interest in our cancer journals was low.
I just want to add one other technical point that actually is addressable. Right now, if you have a BAM file, does anybody know where you put that? Well, I can tell you where you put it. It goes to dbGaP, and you need dbGaP and that's CG Hub. I'm sorry, I'm drawing. There is a process for submitting this into the public domain. We have a paper that's in review right now where we have spent at least 1,000 hours trying to get, because of consents and other formatting issues, these BAM files into the public domain. It is not easy, and there was recently a sort of consensus statement on biomarkers or data availability for our NCI-funded grants, saying that we need to make all this data available. Good luck, because the way it is right now, hire another couple of people and start now, because it is really hard. It needs to get much easier to get the BAM files into the public record. It is extremely hard right now. TCGA did it, but that was a lot of effort. No one else can use the TCGA infrastructure. We need a way to get these BAM files out.
All right, one last question.
Just quick comment, just to push back a little bit at Neil for the RNA being where the action is. Actually, I would argue that the protein is really where the action is, and essentially defend IHC, although it's an imperfect assay. Also, just a comment on the Wild West issue. I think there's a big concern that this really could be overregulated. That's my greater concern than the Wild West. In particular, tumor content. I think the issue of tumor content is potentially, allowing the perfect to be the enemy of the good. Are we going to throw away samples and not analyze them if tumor content is too low? So I have a question for Mickey in particular. What are you going to do for the samples from the match trial where you've now actually subjected the patient potentially unnecessarily to another repeat biopsy to get a fresh biopsy sample and the tissue content comes back at 20%? Are you going to reject that sample?
No, we'll just have a deviation report. It's a good point. I mean, when you're collecting clinical samples, you do the best you can to get the tumor. Obviously, if there's no tumor there, you've got a problem. You wouldn't want to sequence and report anything. So we'll do our best to enrich. We'll set the bar as high as we can so that we can get consistent results, especially with copy number, which we know is going to be difficult already. We will then take the sample and just write a deviation and be tracking that specimen, because it's not going to be the same as all the rest.
All right, I would like to thank everyone. Thanks the panel, and thank you all for coming. Please take a break.